So our second speaker will be Dr. Sonya MacParland, who is an assistant professor at the University of Toronto's Department of Laboratory Medicine and Immunology. She received her doctoral degree from Memorial University of Newfoundland under the supervision of Dr. Thomas Mishlak, funded by the Canadian Network on Hepatitis C, and completed her postdoctoral studies at the University of Toronto under the supervision of Dr. Mario Ostrowski. She is currently a PI in the multi-organ transplant department in the Toronto General Hospital Research Institute, where her lab focuses on investigating how liver immune dysregulation drives liver disease development using single-cell RNA sequencing.
Thank you very much. It's really exciting to be here today. We were talking about this meeting in the PAC meeting yesterday, and I was a former CanHep-C and NCRTP trainee, and this is one of the meetings that really focuses us on why we're doing what we're doing and whether we're asking the right questions. So it's very great that you're all here today, and hopefully I can get you excited about some of the questions we're asking in the transplant program at Toronto General. So in the transplant program in Toronto General, we're interested in general on the cells that make up the microenvironment of solid organs. And the reason that we're interested in that is because if we can figure out how a solid organ is composed, in a healthy individual, we can take a diseased organ closer to a healthy organ. And in my lab in particular, we're interested in the microenvironment in the liver. And we're all very aware that the liver is involved in digestion. The liver is involved in blood clotting. The liver is involved in clearing toxins, fighting infections. And what's really impressive is the liver has a really amazing regenerative properties. And we know that if we end up, unfortunately, with end-stage liver disease, surgeons can take out our liver, put a new liver in, and we can go home. But it's very interesting, though. We can do all these things, but we really don't know the cells that make up that organ. And so in my lab, we're really interested in innate immune cells that make up the organ. And so today I'll be talking a little bit about macrophages. And the important thing to know about macrophages is they're the eaters of the liver. They're picking up antigens. They're clearing things that come through your gut into your liver. But they're doing that without causing too much inflammation in the liver. So under normal circumstances, the macrophages will pick up antigens, and then they'll just tell the other cells that are in the liver that actually everything's fine. We've got this under control. But in the situation where you have liver disease, that balances off. So for example, in hepatocellular carcinoma, which is cancer that develops after hepatitis C infection, there are macrophages that are actually inappropriately quieting the immune system. So you're supposed to have a T cell that comes in and kills that tumor, but the macrophages are actually telling the T cells, actually, there's nothing going on here, go home. So we really want to look at the balance of those cells, and what we want to first do is really describe them in healthy individuals and understand what they're doing in diseased individuals. And so there's been a lot of beautiful papers that have described the molecular biology of the liver, but up until now, we've kind of described the signatures in the liver, so that's what the cells are doing, what they look like, what they're secreting, as an average of whatever's happening in the liver. So you would take a liver biopsy and you look at what's the overwhelming signal in the liver. So you're almost looking at the liver as a smoothie. And you know, these beautiful papers have shown that there are clearly distinct populations within the liver, and these cells respond to the microenvironmental cues in the liver. So based on whether the liver is sick, whether there's appropriate blood flow. And, you know, through this program, actually, there have been some beautiful papers that show that there are cellular signatures within the liver that are associated with responses or non-responses to hepatitis C therapy. But we really need to know the knowledge of the individual cell populations in the liver if we want to go in and start reprogramming livers to even prevent the need for transplant, for liver transplants. And so what we want to go from is the description of the liver as kind of a smoothie to the description of the individual fruits that make up that smoothie. And so the way we can do this is using something called single-cell RNA sequencing. And so what I'm going to describe is this really simple study that we carried out to generate a comprehensive cellular map of the human liver. And so what we wanted to do is do this to make a foundation for single-cell disease studies. And simple questions have sometimes it's a difficult path to those questions. And usually for liver researchers, it's through the tissue. And so in order to acquire really high quality tissue, we went to our beautiful transplant program at Toronto General. And in that program, they removed something called the caudate lobe during the transplant procedure. And what we could do with that lobe is we could cannulate it with its own vasculature, so its own blood vessels. And so we could just gently remove the cells from the liver and flush them out. So any of the cells that are circulating, because what we're interested in is the cells that are actually sitting in the liver and causing disease. So once we flush those cells out, we could introduce something called enzymes, collagenase and DNase, neutral protease. And what we could do is gently disrupt those cells and break apart those hepatocytes. And then we could just release them from that caudate lobe. And what's important to note here is that we did no mechanical dissociation. So the most important thing is that hepatocytes and endothelial cells are very responsive to stress, like chopping and smushing and breaking up of the cells. So we did no mechanical dissociation, and so we ended up with a total liver homogeneity. And so what we can do then from this soup of cells from the liver is we can actually take each individual cell and we can sequence all the transcripts or all the RNA in that cell. So what we can do is find out from each cell what they look like, what they're how they're activated, and we can use that to infer what they might be doing in health or disease. And so I'm going to show you something called Tisney plots today. And what that is when we have this information from each of these fruits that make up the smoothie, we then cluster that information so we can group cells that seem the same. So then we can see about the frequency of cells, we can see what they're expressing, and then we can compare them to each other. So we take those cells, we cluster them together in a Tisney plot. So that's just taking this complex information and clustering it in a 2D space. And then we can compare and pull out those populations and compare them to each other. And so, and you can compare them within a patient, but you can also compare them between patients. And so using these methods, our team developed a single cell map where we took 5 healthy livers and we sequenced 8,444 single cells from these 5 healthy livers. And this actually revealed to us that there are 20 clear populations of cells within the human liver. And there was things that we knew we would find. So we found different populations of hepatocytes, which we expected. And we found a population of cholangiocytes, and they're tricky cells to pull out of the liver because they're hard to break up in this dissociation. And the cholangiocytes are the cells that make up the bile duct. And then we saw, actually, it was interesting, we saw different populations of B cells, both that are producing antibody and that are ready to respond to antigens. So mature B cells and plasma cells, the antibody-secreting B cells. And then we saw a very nice, or several very nice clusters of different types of T cells. So these are the cells that would be responding to the tumors, responding to viral infection. And there are quite a vast number of T cells actually that are resident after you flush those livers and get all that blood out. We also found different populations of endothelial cells. So these are the cells that line the sinusoid. So we can look at how, the endothelial cells are involved in disease. And what we can do, and I won't go into too much detail in this slide, but what we can do with this technology is we can take the single-cell RNA-seq data and then find antibodies that correspond to those highly expressed genes, and then we can look at where the cells are found or zonated in the sinusoid. And the important reason to ask that question is because you could have the cells present in the liver, in a diseased liver, but they might not be in the right area. And so then they can't be really doing their job, because we know from the liver that there's a very distinct division of labor within or across the sinusoid. So it's important not just to look at the transcripts, but you have to see what the cells are doing, what they look like, and where they are in the liver. So this is spatial analysis. So what we're able to see is that clusters that we found that were distinct actually were found in the central venous area of the sinusoid. And then we're very interested in macrophages, again, because of their clear role in disease, but also, you have lots of macrophages in your body, but per organ or in your liver, you have the most macrophages of any solid organ in your body. And so we wanted to look at those macrophages in terms of what they were expressing. And what you can do is actually, when you look at the genes that they're expressing, and these genes actually aren't important, it's what they all kind of encode for, you can ascribe an identity to these populations. So for example, these inflammatory macrophages are expressing lots of different genes that are involved in the inflammatory response, like calprotectin, like lysozyme. And then the second population of macrophages that were characterized by the expression of MARCO were expressing genes that correspond to inhibitory molecules. So they're dampening the immune system. So they would come into play when you have HCC and you have the T cells that are not responding. These macrophages could be prevailing. And so what we wanted to do, again, seeing the cell identity is just the first step. You have to see what these cells are doing. And we wanted to look at how these cells differ functionally. So even if they have a different identity, you have to challenge them with the same kind of damage signal or danger signal. For example, we stimulate them with LPS, and then we look at the inflammatory response they mount after stimulation with LPS. So the readout of this is tumor necrosis factor alpha. That's a factor that's secreted by macrophages that are trying to bring cells in and trying to activate an inflammatory response. And what we saw when we looked at the total cells, we looked at single live immune cells and these cells that are macrophages. And when we separated these cells by either the fact that they're expressing something called MARCO, which just differentiates an inflammatory versus a non-inflammatory macrophage, we did actually see that these non-inflammatory macrophages actually produced only 11% TNF alpha, whereas these inflammatory macrophages that we think are inflammatory because the genes they're expressing, they were secreting 27.7% TNF alpha. So it suggests that not only do they look like an inflammatory macrophage, they actually act like an inflammatory macrophage. The other really neat thing we could do with our computer science colleagues and our bioinformatician colleagues is you can take the genes and look at what pathways they encode for. Say, for example, 10 genes you're expressing, that probably means that you're doing this. And so what was interesting is we could use that to confirm our findings from our in vitro assays and our literature searches. And what we saw was that the inflammatory macrophages were expressing or had active pathways in actually the innate immune response and bacterial defense. And then the non-inflammatory macrophages that were producing little amounts of TNF alpha actually did have active pathways in suppressing cytokine release and in inhibiting myeloid differentiation. So again, it just reinforces the fact that these macrophages not only look different and act different, but they probably are different. And so this brings us to a model of the liver where we can start to pull out the different populations within the liver, and then we can start to see where they are, what they look like, what they're doing, so that we can use this as a framework to look at diseased liver. So what's next? So what we're trying to do right now is we're trying to expand the map beyond 5 individuals. We've gotten to 23 livers. And the reason that we're doing this is because we want to look at the liver over the course of life, because we know there are some diseases that affect children more. And we also want to look at it over the course of, we want to compare women to men. There are diseases such as primary sclerosing cholangitis, which affects men more than women. And so you can't have a map of men and women to just look at, if you want to look just at men. So you want to have a separate female map, a separate male map, ideally a separate child map, a separate adult map. So we're trying to look at this as across life and amongst genders or between sexes. And then what's really neat about the transplant program, one of the neat things about the transplant program is that they're very innovative in the way that they care for patients who have end-stage liver disease. And so recently what they've done is they've taken a hepatitis C positive patient who had end-stage liver disease and there wasn't very many organs available, and they actually transplant in hepatitis C positive liver. And so this patient, this hepatitis C positive liver actually there's no fibrosis, there's no cirrhosis. This is a normal liver. So this patient is early in their chronic infection. And what's unique about this is that you can actually see how hepatitis C impacts the liver without having the noise of fibrosis and cirrhosis involved. And so what they do is they transplant this organ into the positive recipient, they let the patient recover from the surgery, and then they treat them with DAAs and cure them. So this patient still goes home and is absolutely fine. So what we did was we pulled our 5 healthy livers from our original map and we compared it with our hepatitis C liver, our hepatitis C infected liver. And what we saw was pretty neat. The hepatitis C infected liver, when we clustered it with the 5 healthy livers, was very distinct compared to the 5 healthy livers. And I'll just bring your attention to cluster 13 and 7. So these are those 2 macrophage clusters I spoke about earlier. And so the coloring of this map is based on what patient contributed to each cluster. So what you'll see is if it's only one color, that's one patient. So this is the hepatitis C infected patient. What's interesting here, what you'll see here is 3 and 7 are the 2 macrophage populations. So this is the immunoregulatory macrophages, and these are the pro-inflammatory macrophages that are mediating inflammation. And so what you see is they look almost like a rainbow. It's because all 5 patients contribute to those 2 clusters. So there's a lot of, it's not like each patient has a different cluster of macrophages. But then, of course, these 2 populations are differentiated, and they're expressing genes that are either inflammatory or non-inflammatory. So when we looked in our hepatitis C infected liver, this was the only cluster of macrophages that we saw, and actually the one cluster of macrophages in the hepatitis C infected liver was more immunoregulatory. So this is something that we're pursuing, but it may suggest that, the macrophages, because of the fact that there is a dampened immune response during hepatitis C, this may be, the macrophages may be involved in that. And so we're trying to produce this data and provide it to as many people as possible because we can't answer, the data itself can be the platform for many liver diseases and even comparing between organs. So we've made this data publicly available through the Human Cell Atlas program. This is a really great global initiative that Toronto is leading, or the Toronto General Research Institute is leading in terms of the liver part of this map. And so this data is publicly available. There are a lot of really great tools you can use actually to go in, even if you're not a bioinformatician, take your favorite gene, take your favorite cell population, and you can just take the data set and play with it like you would in your, with your flow cytometry data. So, and they actually have multiple organs, if you're looking at gut, if you're looking at kidney. And then, what we're looking to do is translate these results beyond hepatitis C. And so what we want to do is ask some cool research questions. First, the question is, can we target immune cells to improve liver regeneration? And this is a question that's being asked by Michael Cheng in the lab. And what he's doing is he's generating mouse maps of single-cell RNA-seq mouse maps. And the reason he's doing that is because in a mouse, if you cut off 90% of the mouse's liver, it'll grow back. But if you remove the macrophage populations, that animal won't survive. The liver won't regenerate. But it's really not a clear, and this is known, but there's not a clear understanding of which macrophages are required for this. And if we can pull that information out, then you could take a patient who has chronic liver disease or like end-stage liver disease and add some of those cells back in to try and promote regeneration on a disease background. And what he's seen actually is that there are these 2 populations also in mouse. And then the second research question that's being asked by Louis Liu, and he's funded by the CanHepC with our wonderful colleague in Memorial and my former PhD supervisor, is how can we selectively target and repolarize immunoregulatory macrophages within the tumor microenvironment in a spontaneous model of hepatocellular cancer development. And this is using the woodchuck hepatitis model, and this is using some lovely woodchucks. And so to that end, he has generated a single-cell map of the woodchuck tissue. And this is actually really neat because this is a very difficult model to work with because of the fact that there are very few molecular biology tools. So this is a really cool way. Not only can you use single-cell RNA-seq to expand our understanding of human liver, this allows us to leverage new models for looking at liver disease. So I'd like to acknowledge, this work isn't possible at all without a huge team, and we talked about this a lot in the PAC meeting yesterday, coordination of the research coordinators, of the nurses, of the fellows, of the surgeons, of the bench scientists, and people understanding how those livers actually, how quickly they need to be processed. So it's a huge team effort, and so I'd like to thank all the members of my lab, of Ian McGilvery's lab, of Gary Bader's lab. Gary Bader has a very impressive bioinformatics platform. And of course, I'd like to thank the funders, and most importantly, I'd like to thank the the CanHEP-C, because they have supported me through my grad school, and kind of guided me towards a career in liver disease. So I'm very grateful for this program.